Welcome, decoders. Today we will be decoding the history of DNA. Let us make it simple and clear. Erwin Chargaff, an Austrian biochemist, studied the chemical composition of DNA between the years 1949 and 1951. He concluded that DNA was made up of four nitrogen bases: adenine, thymine, cytosine, and guanine. He discovered two rules. The amount of adenine will always be equal to the amount of thymine, and the amount of cytosine will always be equal to the amount of guanine. In other words, the amount of adenine will always be the same as the amount of thymine, and the amount of cytosine will always be the same as the amount of guanine. This proved DNA had a pattern and that the bases are complementary. Rosalind Franklin, a female British scientist, studied DNA using X-ray diffractions between the years 1951 and 1953. She produced the famous Photo 51, which showed that DNA has a helical spiral structure. Her work provided evidence about the shape of DNA, the double helix shape, and the consistent width of DNA, the thickness of DNA, because we know that DNA has two strands, so the thickness measures the distance between the first DNA strand to the second DNA strand. What exactly is X-ray diffraction, decoders? X-ray diffraction is a scientific technique used to study structures by shining X-rays on them. Maurice Wilkins studied X-ray diffraction techniques like Franklin. His work supported that DNA has a helical structure. He later received recognition for his contribution, an award for his X-ray photograph. He worked on this between the years 1950 and 1953. And then you also have James Watson and Francis Crick. They built the first accurate DNA double helix model. They discovered DNA has two strands twisted into a helix. Bases pair specifically, adenine gets paired to thymine and cytosine gets paired to guanine. They also explained how DNA can replicate and carry genetic information. They also won the Nobel Prize in 1962. The years that changed science forever were 1951 and 1953 for James Watson and Francis Crick.